Hi, this is Miss Litton, and this is my wonderful period 4 honors biology class. Say hi.
Hi.
And we are going to be discussing metabolic pathways and enzymes, and then we'll also talk about redox reactions as well. So the first thing I want to introduce you to is this picture. Did we look at this picture last class as an introduction? No? Okay. This is small insight, just a little window into what happens inside one of your cells. All of these reactions that you see going on within this is catalyzed by an enzyme. So every single step, for instance right here in the citric acid cycle that I'm showing you, every one of these requires a different enzyme to make it work. And some of the things that you see right here, it just says glycolysis. There are actually 10 different steps in glycolysis and 10 different enzymes. So when I talk to you about the reactions and when we study cellular respiration in our next chapter and photosynthesis in the chapter after that, you're getting a much abbreviated version of reality, okay? But what I want you to appreciate and why we're talking about enzymes is their importance to make all of these reactions work. Now, enzymes are a type of catalyst, and if you just watch this little video of it, you can see that this catalyst, we have these 2 green spheres landing on this catalyst, and here comes some yellow spheres also landing. And the catalyst is going to facilitate this reaction. You'll see a little change, and they go away. Now, a catalyst could be something like iron, like hemoglobin inside your red blood cells, helping you to hang on to oxygen temporarily. If it is an organic catalyst, then it is called an enzyme, and they are facilitating that reaction. Now on enzymes, most of the time enzymes are proteins, but there are things like ribozymes, and those types of enzymes are made out of a nucleic acid, RNA. But for our purposes, most are made out of proteins. Now what are all proteins built out of? Amino acids. And do you remember when we talked about primary, secondary, tertiary, quaternary structure? And we used the pipe cleaners and bent them around, right? Okay, so a protein structure is critical to its function. And here's one reason why. The substrate that binds onto this enzyme, this right here is called the active site. And that's where the substrate is going to bind in order to facilitate that reaction. If you change the shape of the active site, then you lose its functionality and its ability to work. In your lab, you're going to start next class. The enzyme that we are going to be using, enzyme's name is catalase. And your substrate is hydrogen peroxide. Now remember your Ed puzzle, your Bozeman? Talk a little bit about that. Okay, so what catalase will do is it'll bind to that substrate, hydrogen peroxide, and the product that it generates is water and oxygen. Now, what color is catalase? Oh, sorry, let me back that up. What color is hydrogen peroxide? White. Clear. Okay. What color is water? Clear. So if this reaction took place, you're like, oh, a clear thing going to another clear thing. And by the way, catalase is clear. Oh, yay. But one of the products here, oxygen, can exist, right, as what state? Gas. And what do gases do? They bubble. So you'll be able to see that that reaction is occurring because there are oxygen bubbles. Now we are not going to count bubbles for our reaction. We are actually going to use probeware sensors that can detect the amount of oxygen in parts per million, and you will measure then the rate of this reaction based on how much oxygen is generated. Okay, so, and that we'll talk about the lab more at the end. Take a look here, and blue, explain what you can tell about the active site for this enzyme. Go ahead.
So the active site is the shape.
So the substrate is able to fit in as seen.
Primary substrate fits in the active site together.
You're a really great person. I saw the person. Did you see the person? Do you see people in clouds?
He's going to ask a question.
Okay, hang on one second, and I'm going to answer that question. Now, In this case, this active site, did you see it change its shape a little bit? Yeah, it changed its shape. And once it releases the substrate, whatever product it's making, it'll go back to its original shape again. And enzymes can be used over and over and over and over again. Now, I'm going to pretend that you are all enzymes. Okay? And your job, if I released you upon the school, is to go and find people who are by themselves and make them hold hands, hook them together. And you would run around joining everybody's hands together, as many people as you could find. Okay? Now, what would happen to the rate of that reaction if you went now versus lunchtime?
It would be slower.
Slower when?
Now.
Now. Why is that slower now than at lunchtime?
Because there's less people.
There's more people. Who are the people in our analogy right here? The people would be the, you're the enzyme, the people are the substrate. Good. Now on you, where is your active site?
The hands.
Your hands. Your hands are your active site. Now, if you're holding onto your cell phone in one hand and your Caesar lines in the other hand, are you able to do your reaction as well? No. And something else in the active site is called a competitive inhibitor. And if it's bound on there, then your hands are not available to do work. Let's say your bio buddy has a glass of hot tea because it's chilly outside and it's raining for the first time in a million years in Oak Park today. And so they have this hot tea and they trip and spill the hot tea all over your back. Okay? Now, okay, you have this hot boiling tea on your back. Did they pour any boiling water on your hands, the active site? No. Will you change your shape? Yes. Okay? That would be a non-competitive inhibitor. It's binding to a secondary site, an allosteric site, which is changing your shape. All of these things regulate enzymes' activity and their functionality and their ability to do their job in order to generate their product. And that too will be part of your lab. You're going to be able to explore, what do you want to change? The concentration, how much enzyme you have, how much substrate you have, the temperature of your enzyme. Do you want to dilute your enzyme out and see if it works as well? Do you want to add a little vinegar in and change the pH? Okay, thank you. And change the pH. Or a little bit of demand a heart. I demand that you love me. Or do you want to add a little baking soda in there? And that, as a team, is what you're going to be able to decide for your lab. Okay, so we'll keep going. You had a question. Ask me your question. You figured it out? Okay. Who explained this picture? Blue. Slate, this is your picture. Go ahead, Slate. Now, when you're an enzyme running around, okay, when you're an enzyme running around, you can be used once or multiple times. Multiple times. So, because you can use an enzyme, oh, don't trip, I'm fast. Okay, because you can be used multiple times, again and again and again, you only need a little bit of enzyme to catalyze, millions of reactions. And who is that up there? Who, and this character, Gene Wilder, passed away, right? Sad. What is he holding in his hand? That's right. An oldest bio buddy. Why do I have a picture of Willy Wonka holding an everlasting gobstopper with what we're talking about? Go ahead.
Just like in
all right. And now enzymes, do you remember when we learned about the 4 important organic molecules? There's a final coming up, so let's think about those 4 important molecules. What are they again? Lipids, Protein, carbohydrates, nucleic acids. Good. And we talked about the monomers that build them all. What are carbohydrates built out of? Sugars. Do you remember we talked about monosaccharides, disaccharides, polysaccharides? Okay, one of the ones I talked to you about was sucrose. Remember that? What is sucrose built out of? It's a disaccharide. Glucose and a fructose. Where do you find fructose? Fruit sugars. Good. What was the name of the enzyme that broke down? Remember we learned about hydrolysis, breaking reactions in the input of water. Do you remember the name of the enzyme that would digest sucrose?
Oh, wait. Never mind.
So I thought I knew. Do you remember it? Sucrase. Remember? I probably had you pick your enzyme name. Enzymes are specific to their substrate. So sucrase digests sucrose. Okay? So like my name's Winnie, right? So I'd be Winase would be my enzyme. Tell your bio buddy, what is your enzyme name that would digest you? Good taste. All right? Good taste. So enzymes are named for the substrate that they work on. So lactase would digest what? Lactose. Lipase is digesting lipids. Okay? So they're very specific. They're not going to be able to digest something else. Okay? Just like when we learned about, oh, this makes me think about other proteins that are specific, maybe proteins that are found in a membrane. Remember the different functions they can have? What are they? Channels, carriers, recognition, receptors, enzymatic junctions. Good. Those proteins were all specific. They're all specific to their shape. And so these proteins, these enzymes are also specific to the substrate that they will work on. Okay, let's fill in our notes up to that point. Go to where it says basics. Metabolic pathways, of which I showed you some right here. Metabolic pathways are an orderly sequence of linked reactions. An orderly sequence of linked reactions. And it says each step is catalyzed by a specific what? It's in your notes. Enzyme. Catalysts, and I showed you in general, you could have a catalyst, are substances that can increase the rate of a reaction without being altered. The rate of a reaction without being altered. Okay? And enzymes are organic catalysts. Usually a what? What are they built out of? Protein. And it can increase the reaction rate by more than 10 million times. Now think about this. If you have DVR on your TV, some of you when you're rewinding a show or fast forwarding, it can go 4 times, maybe 8 times. It's doing it 10 million times faster by having that enzyme there. Much more likely to occur. Now if you just put hydrogen peroxide out and let it sit there, it could break down on its own, but it's going to happen at a much what? Slower. Yeah, much slower pace. All right. And then substrate, I already gave you that definition, but Blue wants to reinforce what it is. Go ahead, Blue. All right. And then active site. An active site is the part of the enzyme that binds temporarily with the substrate. It binds temporarily with the substrate. And B, any factor that alters the active site, like temperature, pH, an inhibitor, can change the shape of the enzyme and its functionality. Can change the shape of the enzyme and its functionality. Enzymes can become denatured. Now I will give you a little hint right here. You want to highlight that B, that whole B, because when you do your lab write-up, you will want to refer to that information right there. Okay, that will be, so you could star it and say lab or something. Okay, and then we said that enzymes are not consumed by the reaction, so only a small amount is required. Only a small amount is required. And then the last part is about the naming, and Slate wants to remind you about the naming. Go ahead, Slate.
But it could be like you die
Yes. No, they can build too. Synthetic. All right. Now, would you classify this, tell your bio buddy and support with evidence, would you classify this as an exergonic reaction or an endergonic reaction? Make your claim and support with evidence to your bio buddy. Overall.
It makes them
Remember, you're talking about delta G. Overall, is your delta G going to be negative or is your delta G going to be positive? That's what you're trying to think through. Look at the starting point. Look at the finish line.
That guy is like crazy. It's like the same size as
All right. Exer or ender? Good. Because your starting point is here. Your ending point is here. So this one had more energy. This one has less energy. So our delta G is going to be what kind of number? A negative number. Our delta G will be negative. However, what's the however you need to tell me? You have to start off by putting some energy into it initially to get the party started. And in reference to this, I would talk about a fireplace. Did I already talk about a fireplace with you? Okay, so here's a fireplace. If you have a fireplace and you have, because it's rainy, you know, you want to make some brownies or some cookies and watch a movie later today, right? I said you put all those things in that fireplace, right? Your twigs, your paper, your wood, and then we want to burn it, right? But we don't worry about it spontaneously combusting in our fireplace when we walk by, the blink of our eyes. What do we have to do? We have to light it. We have to invest a little bit of energy initially. Then that will provide enough energy for the next and the next and the next until it's burning down and we have those ashes in there, right? So this is referred to when you talk about metabolic reactions as the E sub A, the energy of activation. Okay? Enzymes play a role in lowering that. They're not going to change the starting or the final position, but they will lower the hurdle to make that reaction more likely to occur. Dark shirted togaed person. This is yours. Light shirted is going to get a chance in a minute. Just dark, this one's yours. Go ahead, explain this diagram. On the outside. Dark is on the outside. All right. Come back to me. Questions?
So is it possible for a reaction to need more energy than it produces? Like if it's exergonic, but it requires more energy than it produces?
No, then you're probably talking about an endergonic reaction, and endergonic reactions always require an input of energy. So you don't have to worry about those happening spontaneously. That's usually not going to happen. All right, now, next, who is it? Light shirted one. This is your diagram to explain. Go ahead, light shirted one.
the energy of activation is really high.
And when the enzyme is present,
Okay, come back to me. Yes.
Wait, I have a question. As an example, would salt be an enzyme? enzyme? Like if you're adding salt to boiling water, does that make sense?
Okay, so what you're doing there, remember when we learned about water, what kind of bonds it likes to make? Hydrogen bonds, right? And the more hydrogen bonds, remember, has a high heat capacity? Okay, so when you put salt in there, then you're making that water be bound water, and it hooks up either with the sodium or the chloride. So it's going to boil at a hotter temperature. That's why you're doing that. Okay. All right. So let's go to our notes on that. Okay. And enzyme substrate complex. No, I did that right. I'm on energy of activation. Yeah. Okay. Defined as the energy that must be added to cause molecules to react. The energy that must be added to cause molecules to react. Why do we have this little hurdle? This hurdle of energy prevents molecules from spontaneously degrading. Prevents molecules from spontaneously degrading. It prevents exergonic reactions from happening without any what? What do we want to have? Controls. Without any controls. And blue, tell them number 3.
Wait, what am I doing?
Okay. Enzymes speed up reactions by lowering the inherent energy.
So that's how enzymes work. When we look at that series of metabolic reactions, that's pretty complex. And I'm going to tell you, if you just add heat to this system, what kind of energy will increase if you add heat to the system? What do we call that energy? Kinetic. Kinetic energy. Wait, who said that? Did you say it? Awesome. Kinetic energy. And the thing about kinetic energy or heat, it can make all of these reactions occur. It's not specific. Enzymes will make this specific reaction occur. And if you don't have that enzyme, then that reaction doesn't occur. Okay? So regulating enzyme functionality, that's not like, oh, that mean inhibitor, he's not letting that enzyme function. It usually serves a purpose, okay, to turn enzymes on and off, to turn their functionality on and off. Now, Sam asked a question earlier about, do they always break down? No, there are other types of reactions as well. So I would like Slate to take the top picture and Blue to take the bottom picture. Go ahead. Okay, I think you can handle that. All right, now let's talk about some of those factors that regulate enzyme activity that we mentioned up here in B, which you may want to consider for your lab. Okay, one of those is substrate concentration. Now I asked you, when would you be able to join more hands? Right now or at lunch? And you told me when? Lunch. Why? There's more substrate, right? There's more people. So as you increase substrate concentration, you would assume your rate of your reaction would increase as well. But at some point, it starts to level off. And the explanation of why it levels off is right in the middle of this graph. And light-shirted bio buddy wants to explain that. Go ahead, light-shirted bio buddy. Where are these located? Yes, this is on that enzyme. There's an active site, and if all the active sites have something on them, then you can't move any faster than that. Okay, we have, whatever, 30 people in the room. If all of you are joining hands, you can't go any faster than each of you joining those hands. Are you with me on that? Yeah. So it doesn't matter if there's 1,000 kids out there or 150 kids out there, we can't make this reaction go any faster. Okay? Another thing that can affect the rate of a reaction is temperature. So there are each enzyme has some peak temperature where it works best at. It may be in these colder temperatures. The reason why the enzyme is not working is because at those colder temperatures, molecules might be moving what? Slower. That could be part of it. And now they're moving faster, so we get the most action. Why does it start to just fall when we hit the temperature? Yeah.
Because they're moving too fast.
Okay, give me something else. What do we, yes,
They become a gas.
looking for something else. Think about the enzyme. What is an enzyme made out of? Proteins. What can proteins do when you heat them up?
Denature.
Denature, right? And the example I would give you is if you take an egg, right, and you crack it on a hot pan, initially the white of the egg is actually what? Clear. But then as you add the heat, it becomes white because you've denatured that protein. If you used a flat iron on your hair today, you denatured the proteins of your hair. That's going to be a temporary because it's going to go back to whatever it was before. You can chemically put a straightener on your hair and that will last even longer. Just like you can chemically get a perm and make curls in your hair that last a longer amount of time. Are you with me on that? Okay, so proteins can be denatured. They can change their shape. Yes?
What's an example of a reaction that works really well with low temperature?
Anything that survives in a low temperature, well, that it would be fine. Any bacteria that lives in really cold conditions. If we talk about animals, like who can live well at the South Pole? Could you have, or in Antarctica? Could you have a polar bear there, or could you have a lizard? Polar bear. Why? Yeah, so the lizard's not adapted to that cold temperature. So any organism that lives where it's cold, they're adapted to that. What were the extremophiles? A whole group of bacteria that live where the pH is really low pH or it's really hot. Yeah, archaea. Remember archaea? Okay, and like some are thermoacidophils. They can handle that. Can you and I live in those conditions? No. So different organisms have different adaptations for that. Okay, so if you look here, this would be my example. So if those runners that we have running right now, they're going to run in the mud, right? And good luck to them. But if you're a runner running around Oak Park in your chaparral environment, okay, when would you want to run into a rattlesnake? At 6 rattlesnake. I snack on them all the time. I have tried rattlesnake. Has anybody ever tried rattlesnake? Yeah. It tastes like chicken. Okay, so when would you want to run into a rattlesnake? At 6 in the morning or 12 noon?
12 noon.
6 in the morning.
I want to go and run into them at 6 in the morning. Why?
Because they're not heated up.
They're not heated up yet. They can't maintain their body temperature, so they won't be able to strike me as fast. 12 noon if it's hot outside. Okay? What about all the people?
Surrounding me to protect you.
Oh, I didn't know. I was by myself. Okay, so body temperature of ectothermic animals limits their reactions unless they're at the right temperature. Now, if you're exposed to a polar bear, as I mentioned earlier, it doesn't matter if it's 6 in the morning or 12 noon, his claw can still wipe your face off your skull. Okay? Okay? Done. Okay? So they're an endothermic animal. It maintains its body temperature, so all of their enzymes are functional. Now, Siamese cats, they have an inherited mutation, and so depending on the temperature depends on whether their enzymes are functional or not for their coloration, and that's why they have their different colors in different parts of their body. Now, just like that, pH can also affect enzyme activity. Here are 2 enzymes that digest proteins. One works in your small intestines, and one works in your stomach. Which one do you think works where and why? Tell your bio buddy.
Trips in your stomach. Think in your stomach.
Where do you think pepsin works? Stomach. Why? Because you know in your stomach the pH is very what? Low. Okay? Trypsin and chymotrypsin work in your small intestines, and they need a pH that would be slightly basic in order for them to function. Okay? And this is why you have like Pepsidase C, Pepto-Bismol. It's all coming from that. Okay? Now, so they work at a particular pH. Now, cofactors and coenzymes. Cofactors are anything that's going to make that reaction, you know, it's like a catalyst, right? It's something that helps an enzyme work better. Helps an enzyme work better. It could be a piece of metal of some sort. It could be like heme, like iron, that's in your red blood cells. Okay, that would be an example then of a cofactor. A coenzyme is something that's organic, but it's not a protein, right? Because enzymes are proteins. So if it's just another protein, it would be a protein on a protein, so the whole thing's a protein. Okay, a coenzyme is like a vitamin. It could be, what are some of our other organic molecules in our proteins? Name 3 more organic molecules. Lipids, carbohydrates, and nucleic acids. If it falls under one of those, then that would be called a coenzyme that would make it function. Okay. Now, I talked about this earlier with you when you were the enzyme, and we talked about competitive and non-competitive inhibition. Okay, and I would like Slate to take A and Blue to take B. Go. Okay. So what does a competitive inhibitor do?
It binds.
Prevents the substrate from binding by itself, binding to the active site. You're competing for the active site in a competitive inhibitor. Okay? A non-competitive inhibitor, it is not binding to the active site, it's binding to a secondary site, and this is called the allosteric site. Okay? This secondary site is the allosteric site. And by it binding here, it causes within that enzyme, within that molecule, to rearrange bonds. So whereas the active site was this shape before, now it's this shape. So the active site has changed. The active site has been changed. It didn't bind to the active site, but a consequence of it binding to this allosteric site triggered this, and it changed the active site. Yes?
So it kind of like pushes it upward.
Well, in this case it did, but it wouldn't always. It's just changing the shape. And remember, enzymes are always very specific because it's a protein. So you change it at all and you change its functionality. Take a look at your notes on that. Go where it says enzyme inhibition. This occurs when a molecule, the inhibitor, binds to an enzyme and decreases its activity. Decreases its activity. Non-competitive inhibition, the inhibitor binds to the enzyme at a location other than the active site. That site, name it. It's allosteric. Allosteric. And that would be a form of negative feedback. It's shutting that enzyme down. Allosteric site. Competitive inhibition, you have it all. So Blue, just tell them what it says there for competitive inhibition.
Yeah. You're telling me about competitive
All right. Now, as I said before, inhibitors aren't always bad. If you don't need more of something, don't spend time producing it. Don't waste the energy or the molecules making something that you don't need. Now, right here, this is showing you a metabolic pathway. A becomes B, which becomes C and D. These are all different steps. And then you have your final product F. Each one of these steps requires its own unique enzyme to make that product. This first one right here, here's enzyme 1. Here's your first reactant, A. What would you call this right here?
Active site.
Perfect, the active site. Now, it goes, and now you have a whole bunch of this product F. You don't need any more. So you want to turn it all off. This is very fiscally responsible. It doesn't turn off the enzyme right before it. It goes all the way back to the beginning. And the very first enzyme, if you look right here, what site would this be referred to where it's binding? The what? Allosteric site. It's binding to the secondary site. It's not binding to the active site. It's binding to the allosteric site, but what has happened to our active site? It's changed its shape. So now substrate A can't bind, so it shuts the whole series of reactions down just by controlling the first step. Conversely, when you start to run out of F, it won't bind to the allosteric site any longer, and as a result, the active site becomes back to its original shape, and it'll facilitate that reaction again. Not it. Now stop. Each of you is going to explain this right here. One of you is going first, one of you is going second. Not it decides who goes first, who goes second. Go. All right. Birds of Oak Park, welcome. And remember, this is a map. 142. And what is 142?
How many
Families of birds are, yeah.
I have like 4 words.
Yeah.
Duck,
Hawk.
yeah.
Oh my god.
Vulture.
I totally meant to put that.
Parrot.
There we go.
Toucan.
Oh, yeah.
Parakeet.
Hummingbird.
Oh. You got 10.
We're out of control.
12. An owl. Is an owl?
I know, yes. An owl is a bird. Well, I put 13.
Oh yeah, it's
So good at this. I'm a bird master.
I know some bird names just from watching this list all day today.
now I don't trust you.
All right, come back to me. Before I hit stop, let's chat. The active site of a specific enzyme, all enzymes are specific, right?
Yeah.
Is it similar to that of any other enzyme? No. Why? It's specific to its what? Substrate. Yeah, that's what I want to say. Is the part of the enzyme where its substrate can fit?
Yes.
Yes. Can be used over and over?
Yes.
Yes. Is not affected by environmental factors? It is. We would have to cross out the not. Are you getting a gong of victory? Bell of shame. Ring that bell. Look at their clicker. Are you giving them a gong?
You made me get the wrong.
And I'm right behind you.
Good job, Gray, Gnatcatcher, Housefinch, Hermit Thrush. You're in the medal room.
It's definitely D. Oh yeah, that was just a joke. It's a cat.
All right.
That was the point.
I thought.
Wait, are these like people pets or like
No.
birds that
Eagle. 13. Oh, yes.
Gong or bell of shame? Gong. Oh.
Ben, how could you?
Fine. Okay.
They have some mind of their own.
Come back together. Guys, we gotta move on. Okay? Well, here we are. Here we are up here. The diagram below depicts non-competitive inhibition, and most of you said true. 89% of you said true. And you knew that because the competitor did not bind to the what?
Active site.
Active site. The competitor didn't bind here, it bound to a secondary site, which then changed the active site. What is that secondary site called?
Allosteric.
Allosteric site. Okay? Shame or gong?
American Robin. Sounds like Red Robin. Oh, geez. Oh, yeah. Yeah.
All right, so before I hit stop, tell me, is the allosteric site on an enzyme the same as the active site? No, it's two different sites. Is it non-protein in nature? No, it's protein because it's part of the enzyme, right? Is it where ATP attaches? No, did I even say anything about that? No. Is it often involved in feedback? Yes. Oh, look at that clicker. Bell of shame, gong of victory. All right. Okay, and I'm going to go ahead and stop this recording right here. If you're up really late at night, go and make yourself a piece of toast and don't stay up too late.